el marco del curso de programación fetal, contar con la presencia de Pascale, maintenant en français o en anglais, no, en anglais, va a dar la charla, así que adelante, bienvenida.
Gracias. Gracias por invitarme. Siento no hablar español, así que voy a hablar en inglés, pero la próxima voy a hablar mejorado mi español. So, thank you very much for inviting me. So it's a pleasure to be here with you. So I've been asked to talk about the horse programming, which we started a long time ago. I'm going to try to summarize 15 years of work in 2 hours. So it might not, might not cover everything, but it's most of what's going on at the moment in programming in the horse. So I work in INRAE, the institute INRAE, but we belong to University Paris-Saclay, which is now the largest university in continental Europe. So first I will talk to you, I don't know how much you know about it, setting the scene, what is DOHaD, what are epigenetics, and what are the critical periods, and please stop me if you don't understand or if my English is too difficult. Then first, when we started wanting to work in horses, we need to actually have a proof of concept to show that programming was effectively taking place. Then I'll follow with the importance for horse breeding, and I'll finish with concluding remarks. So it all started with Professor David Barker, who was in Southampton, and who was an epidemiologist, a human epidemiologist, who actually showed in the late 80s, early 90s, a number of papers where he was trying to actually relate the the mode of life of people, and especially their nutrition, and if they smoke or not, and lots of things as adults, and relating it to older disease in older people, such as cardiovascular disease and type 2 diabetes. And here is one of the first papers he made. What happened is he was comparing the population near London, which is pretty rich and wealthy and eating pretty well, compared to the people in the Midlands, which is Liverpool, Manchester, where the population was less affluent and was drinking a lot and having a different nutrition. And so he was comparing modes of life and onset of disease. And then he realized that a lot of his population did not move. And in the early 20th century, nurses were collecting the birth weights and the weights at 1 year of age of all children. So he actually could put in his epidemiological work the birth weights and the weights at 1 year of age of the people he was studying. And this is what he came with, that the most important factor, out of all the other factors, nutrition, drinking, smoking, for death from coronary disease in this population was the lower weight at 1 year of age. This is in pounds. And you see that this is the odds ratio for mortality from cardiovascular disease, and it's increased in the children that were of low weight at 1 year of age. Then he worked with Nick Hales in Cambridge, and here we're back in grams, and they looked at perturbation of glucose homeostasis, and here they have the same thing, that is the highest risk, and you see the odds ratio is really increased, is in the children that were born with a low birth weight. And same here, a bit later, he looked at syndrome X, which now is called metabolic syndrome, and the odds ratio is between 15 and 20 for the children that were born less than 2.5 kilos to develop metabolic syndrome as adults. as a veterinarian, I was working with Peter Rosdell at that time, which in the UK, and Peter told me, oh, you know, come, we're going to listen to someone at the Royal Medical Academy, and we listened to David Barker. I think it was around here at that time, and his talk was very controversial because there were pediatricians and people who told him, you don't understand anything, you're talking nonsense, you're comparing something that happened at birth and something that happens later on, you don't understand, and he was really very much attacked. And my boss, Peter Rosdell, told me, Pascale, if it works in humans, it works in horses. And that's how I started to, and it actually was the line of all my career, maybe extending to other animals and also humans. So anyway, up. So now we know, I mean, you know, time has flown. We started with Barker hypothesis. We had no mechanism. I think one of the major points is that Barker said, I observed that, but he had no idea of why it happened. So he made a theory, which I didn't put here, which was Barker hypothesis. And after, with time, we developed the understanding of epigenetics. I was involved in taking care of clones, bovine clones, and they were genetically identical, but they were not the same. And so, which actually really led to the understanding of the role of epigenetics in the phenotype. And that's what happens in programming. So we know now that adverse nutrition or metabolic status or whatever of the parents, so that's including the male, and I won't have the time to talk in the male, there's not very much data yet in males in horses, will induce fetal growth adaptation through adaptation of the transcriptome, the cell growth, the tissues, the organs, and the memory of that is the epigenome. The modification of the gene expression is the epigenome. I'm still wondering how much blood flow, changes in blood flow that are immediate do not precede epigenetic changes. You know, it's something that probably physiological changes are preceding the epigenetic changes, and the epigenetic are making a memory of it, and that stays. So all these changes are aiming at improving immediate survival of the offspring. And there's another term that is important in programming, which is predictive adaptive response, which means that this fetus develops in an environment that is kind of a proxy for the postnatal environment. So basically, the adaptations that are developed here are future adaptation to the future environment, in particular in terms of restrictions. And we call it the predictive adaptive response. And the worst case scenario is what we call the mismatch. If you've been adapted to restriction and then you have plentiful here, then you're not programmed for plentiful. And that will be one more thing that will actually make you develop disease. And the diseases are mainly metabolic disorders, so type 2 diabetes. I put osteoporosis because in horses we have osteochondrosis, and that was really one of the themes of my work. Does OCD in horses, can it be programmed? Obesity and many other diseases. We know that mental disease have some roots also in the fetal life, and you know, it's really developing quite a lot. And maybe also the immune organs are also programmed and may, indirectly also program resistance to infection, but that still needs to be more studied. Anyway, so now we know that it is based on the epigenetic programming. We have a DOHaD society, an international DOHaD society, and this phenomenon is widely recognized. And as we are in a veterinary faculty, yes, this affects all different species, including the horse. So what are the mechanisms? Just, there's almost no data in the horse on epigenetics, on programming, so I won't go over it. I mean, we are currently working on it. But just to remind you the mechanisms, so there's DNA methylation, which is the placement of CPG methylation on CPG islands or CPG bases, and it's usually associated with promoter regions, but not only. Usually methylation will close the chromatin and remove the access to transcription factors. Then we have the histones here, and on the tails you have histone tail modification, which may be acetylation, methylation, and tons of other modifications. They are really increasing, and we are not seeing them all. One of the things that is actually discussed by epigeneticists, last week I was in a vivo of one of our PhD students, and there was a big discussion. Can non-coding RNAs be considered as epigenetics? We do think it's epigenetics because they are affecting the messenger RNA transcripts, but some people say no, because in fact the non-coding RNAs are under genetic control, so they could be, they're both genetic and epigenetic, but they will affect the expression of genes. And of course, it's not so recent anymore, but all this can be modulated by environment and nutrition, especially, particularly in domestic animals, which are of interest for us. So as a summary, here is the open chromatin, where the transcription factors have access to the DNA, and it will be repressed in the epigenetic factors. So you have histone erasers, histone deacetylases, histone lysine demethylase, and de novo DNA methylation that goes through an enzyme called DNMT3 that will actually close the chromatin. And what's important is methylation and epigenetic parameters are transmitted through mitosis, and it's this DNMT1 that maintains the epigenetic factors in mitosis. And then we have other factors that are histone writers, HATs, and TETs, which are doing demethylation that will open the chromatin. And you can have changes like that quite often. So some are labile, that means they will not last for a long time. And some, especially when they are put in the periods, the very early periods, developmental periods, these marks will remain, and that's the basis of the programming throughout life. What's really important for us to know is that there's a lot of nutriments with enzymatic activity that will actually change some. I mean, the most well-known example in humans is broccoli. Broccoli has a HDAC, I think it has a HDAC enzyme in the broccoli. So when you eat broccoli, you will actually change your histone acetylation. But then it doesn't last very long, for about 24 hours, and it comes back to normal. So it has no clinical issue. But a lot of feed have some epigenetic factors. We made a review, and that's also why I'm a bit worried about we're talking about agroecology and changing the nutrition of our animals in order to actually adapt to the changing climate. However, people actually control animal production, but they don't control transgenerational effects. And I think that's one of the things that maybe some of you will study later on. And the other thing is is here, the key role of the methyl groups. Of course, as you've seen in a lot of these things, you have methylation. And in order to do methylation, you need to have methyl groups. So, of course, the food and the quantity, the availability of methyl groups is going to be very important to enable these changes to take place. And one of the particular period that is really important is the embryo. When we have embryo in culture, the availability of methionine will be important and will actually change the programming and the methylation of key genes in the embryo. So that's just to illustrate the mismatch. Here we have the developmental environment, which can be optimal and impaired. and then the adult environment, which can be sparse or plentiful. And it's a good way to represent, this is the areas that we can encounter in our adult life. So if you've been developed in an optimal environment, then you can adapt to quite a lot of conditions, but not so much to very poor conditions. Whereas if you've been developed in impaired condition, then you will actually develop better in poor conditions, but you will not adapt to very plentiful conditions, and that's where you have this mismatch, and there's an increasing risk of disease from this unpredicted excess. So I think that's what you need to remember from this. The other point, still in the basics of DOHaD, is that, as I told you, there are critical periods of development. First, the sperm and oocytes. Initially, we thought, because they're highly methylated, the genes are really highly methylated, so there's little access to transcription factor in the gametes. However, they're not fully methylated. So there are methylations that actually can be transferred to the embryo. Secondly, the sperm is also accumulating, and the oocytes are also accumulating a non-coding RNAs. And the sperm transfers non-coding RNA to the zygote, and depending on the condition of development of the oocyte, you will have also different non-coding RNA. So there's really a focus also on the gametes, and it's still a subject of lots of discussion in terms of epigenetics, but that's when actually epigenetic changes can actually be transferred through the germline in several generations. And also in the sperm, normally the histones are replaced by protamines, but not all histones are replaced. So you still have histones, and they can be modified, and that can be transferred. So then the embryo will develop mainly on the reserves of the oocyte, and then you have genome activation. Then all these cells are totipotent. And then afterwards, you have the first differentiation with the blastocyst. Here, the inner cell mass and the trophoblast. And of course, this is dependent on epigenetic mechanisms. All cellular differentiation is epigenetic mechanisms. So this period is particularly fragile to any modification of epigenetic environment, and that's why the nutrition of the dam can be really important, and embryo production can actually change also methylation and other organs. So then we have implantation, and the placenta is known to be less methylated than the somatic cells. Organogenesis, which is a lot of progress also of epigenetic mechanisms. Gonadal differentiation, which is really important. Placental function takes place. So in my lab, we work in many species, so I've put a number of species that we use. And then birth occurs, and so during all that time, the environment will actually play on epigenetic marks and might disturb some of these processes. And maybe those marks may stay or not. So it's pretty complicated. And then another period where there is a lot of epigenetic changes is the time of birth and the neonatal time, because suddenly the fetus has to adapt very rapidly to eat, to have a valid intestine, lung function, suckling. So it's a period of huge epigenetic changes, and again, we are then in a critical period, because any changes in the environment may actually alter some of these marks, and which may stay. The other period that is really important, but has been really understudied, and is now the object of more studies, especially in humans, is the puberty. Because again, we are in a big change in the body of the individual, and of course, that's driven by epigenetics, and the environment changes may play a role. And that will actually, that will actually drive the offspring phenotype. So this is the basis from which I started about almost 20 years ago to study in the lab. So we started with the fact that in horses, maternal size will determine uterine capacity. We have a diffuse placentation throughout the uterus. So we started from the idea that the size of the uterus would actually determine the size of the placenta, and so the exchange capacity, the contact surface between the placenta and the endometrium, and it would actually determine the nutritional supply to the fetus. And we knew from very old work from Walton and Hammond in Cambridge and Twink Allen, we worked hand in hand with him and all the Cambridge team, that in horses, fetal growth can be enhanced above or restricted below the normal genetic potential for the breed by changing maternal site. So we developed, Twink Allen from Cambridge had developed some models, but he hadn't really looked at effects on metabolism of the horses. And I wanted to know whether metabolism was affected, because he looked only at growth. So here's the first PhD student was Pauline Penier. Here she is with Twink. And so I will show you what we did and also some of Twink's results. So we made normal gestation of ponies. So those are the pony controls. Then we took embryos of ponies and we transferred them in thoroughbred recipients. So we had the pony in thoroughbreds. So we thought that they would be enhanced, larger, because it was a pony and it was in a thoroughbred. And then we had draft mares. So these ones are about 600 kilos, these ones are about a ton. And we transferred embryos in the draft recipients. However, these are about the same size as withers, and probably not. They probably have the same type of uterus. So that's what we did. Oh, sorry. So in terms of placenta, this is the placenta of ponies, and of course we had an enhanced placenta, 1 kilo more in terms of embryo transfer in thoroughbreds, and 2 kilo more in draft mares. So we were quite happy. We did what we wanted. I don't know why the... Yes, so here is the growth curve. So the control ponies are in orange, the enhanced are in blue. So the enhanced are already larger by 57% at birth. And this is, they go until weaning at 180 days. And then you see that they're still 30% bigger in terms of body weight at a year and a half to 2 years of age. And what Twink showed it was that it lasted until 5 years at least, and then he stopped studying them. So we're really changing the growth of these animals. When we looked at the different measurement of the horses, you see that the curves are very parallel, and it seems that the enhanced ponies, the growth is really amplified, at least in a harmonious way, which is important because in humans, this harmonious growth is really associated with most risks. And here we have, so they are enhanced, but in a very harmonious way. And even cannon bone and the front legs here, you see the significance is consistent. So Twink looked at, with Abby Fowden, they looked at the barrow reflexes. And so they found similar, so they treated the animals with sodium nitroprusside to actually reduce tension, arterial tension. So they had a similar fall in the groups, similar increase in the heart rate, but they had a higher secretion of catecholamines in the pony in thoroughbreds compared to ponies. This is the mean arterial pressure, then it was increased in the pony in thoroughbreds, but you see it seemed to increase, but it's not statistically significant, and otherwise they had a similar fall in the pressure. So there is still already at 6 days of age a difference in the pressure response. And so they had also elevated blood pressure here and lower baroreflex. So we wanted to see what happened with glucose metabolism. So in the newborns at 3 days of age, we did IVGTT, and we had a similar glucose response to insulin, to glucose, but we had an insulin response that was higher in the enhanced ponies, definitely higher. So they have a resistance to insulin. These guys, they have a resistance to insulin after birth. However, at 200 days of age, we did the hyperinsulin euglycemic clamp, which is a pain to do. It's an extremely long and tricky exam that afterwards we stopped doing just for sake of time. When we have a lot of animals, it's really difficult. And here we didn't see any difference anymore, and neither did we at 2 years of age or 1.5 years of age. So they have an early resistance to insulin, but then afterwards they look like a normal pony. So that was the enhanced. What happened if we restrict the growth? So we did the same type of experiment. Twink used thoroughbreds, we used saddlebred, we did the embryo transfers in ponies, and we did artificial insemination to do the controls. Which could actually, we'll talk about it later on. It could be, I think it would have been more scientifically good to actually do also embryo transfer here, but honestly, we did not have the means to do it. So what happened? Well, as expected, we have a reduced placental weight, and for the thoroughbreds, it was minus 1.5 kilos, and minus 0.8 kilos in our experiment. And this is our experiment. So in pink, it's the restricted saddlebreds, and these are the controls, and the enhanced saddlebreds, that's the one that were put into draft. But in fact, the uterus didn't really change, so we didn't see very much differences between the ones that were put in saddlebreds or that were put in draft mares. And you see the growth. So the growth is enhanced here until weaning. And of course, the restricted, not only were they restricted in utero, but they get also the milk from the pony mares. So they are even more small. And this here got the milk of the saddlebreds or the draft mares, and they grow. And the draft mares are known to produce more milk, so you see they're slightly higher. But then after weaning, you still have this difference between the groups, no difference between the draft and the saddle, but a difference for the ones that were gestated in ponies, still minus 17%. And what also happened is at this stage, some of these ponies, some of the saddlebreds, when they were born from ponies, they were dismature. So they didn't have to be in intensive care, but they took a little while to get up and suck properly. And then afterwards, you see that the withers height is only different until a little time after weaning. The chest circumference is different there, then it's not different, then it's different again. The front legs are different, and the cannon bone is different, which is a problem here, because if you are, which we are doing, we are raising show jumping horses, probably the cannon bone strength is important for sports. So we're a bit concerned about cannon bone. It was measured with precision measurements. And so, but we don't see very much of a difference between the ones that were raised by draft and non-draft and saddle mares. So basically, because of these that are different, we have a growth of the restricted foals that is slowed until weaning, and then they have a disharmonious catch-up. So in terms of glucose metabolism, again, no difference in glucose response to the glucose under the curve after glucose injection. However, we have higher insulin response in the enhanced animals. So, and then afterwards we have still a difference at 200 days of age. So this is a clamp, so it's the other way around. But basically, these animals are more sensitive to insulin. And then it stabilizes at around 2 years of age. So we were really interested in also whether OCD was affected, because OCD is a multifactorial disease, osteochondrosis in foals. It's a disease that affects foals between 0. In fact, you can actually find foals that have already OCD lesions at birth. So we know that there is some fetal origin. And so we looked at what happened, and we know that one of the environmental factors linked to the onset of osteochondrosis in horses is high glycemia in the herds, high insulinemia. So because we showed that we were actually changing the glucose metabolism, we thought, well, maybe that plays a role. And yes, indeed. In the foals, the saddlebred foals that were affected by OCD, at 6 months of age, 100% of the restricted ones were affected, whereas it was in our population around 25% normal OCD in our saddlebred population. And at 18 months of age, that means after 6 months in pasture, and it's been shown by, especially by the team in Liège at the vet school, that lesions, as long as you don't have the seconds, they can actually go back and forth. They can repair. So we still, however, we still have 33% lesions in the restricted saddlebred, and we have decreased onset in the other groups. So yes, this restriction of the foals can affect the lesions of OCD. So I didn't show you the data for the ponies, enhanced ponies, because it was also a claim in Europe, in France. Embryo transfer was usually done in draft mares. And the breeders were saying that it was increasing the incidence of osteochondrosis. However, here in my model, I used ponies. My ponies don't develop OCD, so they were not a good model. So you could imagine that when you transfer embryos of a normal horse in a draft mare that is going to produce a lot of milk, you might change the metabolism and you might act on OCD, except that in my setup, I couldn't show it. I didn't see anything. So anyway, to summarize, so we showed that there is programming also of the metabolism in horses, not only of the growth. In the enhanced ponies, the mares had enhanced uterine capacity. They had a maternal glycemia in the horses that is reduced compared to ponies, and they have higher milk production. And in the restricted, it's the other way around. Then in terms of fetal growth, we had an increased growth in enhanced ponies and a restricted growth in restricted horses. They had larger placentas here, and when we looked at the placenta, which I didn't show you, there was no functional adaptation. The placenta looked normal in terms of gene expression. At that time, we were doing just candidate genes, but we didn't see any difference. Whereas in the small placentas of the restrict horses, we saw reduction in IGF-2 expression and SNAT2. IGF-2 is the fetal growth factor it's an imprinted gene in the placenta, and SNAT2 is a transporter for proteins, for amino acids. So these guys had an amplified and harmonious growth. These guys had a slow and disharmonious catch-up. And then they were initially a bit more hypoglycemic in the enhanced, hyperglycemic in the restricted. We also had, which I didn't show you, an increase in NIFA concentration in the enhanced, reduced NIFA concentration in the pony, which seems logical. Early insulin resistance, early improved in sensitivity. And later on, they become like their own species, their own breed. So we didn't find any effect on osteochondrosis, but they were ponies, so basically we cannot conclude for enhanced, but we can conclude for restricted. And we found also differences in the thyroid concentration, T3 concentrations between the enhanced and the restricted. So they were like a mirror. So that was really interesting. And so that was, the conclusions was, yes, we need to get interested in that phenomenon and it can play a role in horses. So, yes, that was the end of the PhD of Pauline. There may be truth in focusing on the recipient mares. Not only will she be the recipient of the embryo, but she will carry the foal, and then she will nurture it for at least for 6 months. So we should take care of those mares that are carrying our foals. And the first obvious importance for the industry of this first part of our work was that you have to be careful when you do embryo transfer to transfer the embryo in a breed that is the same size as the breed of the embryo. That's really important because if at that time I was working with my husband on clones, and we were working with the United States, and we had cells from very big French show jumpers or really large, and they were transferring them in how do you call them, the mares you have, quarter horses, that's it. And so that was not done. It was like a restriction. So we had to fight, and finally they actually acquired draft mares to put our horses or embryos into the draft mares and not put them in quarter horses, because I was really fighting, because clones are already difficult to have, and then if you actually make it even worse by putting them in the wrong size recipient, it's not good. But that can be applied also to normal embryo transfer, and so that's really something to take into consideration. The only thing where it can be very useful, it's in ponies. If you have tall ponies that are limited to the size of a pony, and if you put them in large mares, you're likely that your pony is not going to be a pony anymore, and you cannot sell it as a pony. But if you put in a smaller mare, you're likely that you still will be under the limit, and you can actually sell ponies. So for pony competitions, which are big thing in Europe, and probably in the States also, and maybe also in your place, no? Well, in France, parents invest a lot of money in the bodies of their children. It can be really important to actually think of that if you do an embryo transfer. Anyway, so then we went on. We had the proof of concept, so we went on thinking of David Barker and looking at nutrition. So as I said, nutrition in mares can induce laminitis, hyperlipidemia, Cushing syndrome. We surmise that it can actually play also a role on osteochondrosis and insulin resistance and endotoxemia. So we focused on that. And so for osteochondrosis, we know that it's a really multifactorial disease. Genetics play a role, the breed, the genetic heritage, the postnatal environment, biomechanical trauma, mineral imbalance. The copper has been shown to be important, the growth rates, the housing, the exercise, and the nutrition and the carbohydrate metabolism. And we know that we can actually, if you have a diet which is with a high insulin post meal, then you will affect the vascularization of the bone, and you will actually affect the formation of dissecants here in the bone. But the maternal environment plays a role too with the nutrition and the carbohydrate mechanism, and we can affect that in horses. And the first work that we did was with the team of University of Liège in Belgium, because they have a group that is really specialized in OCD. And I met the head, Didier Certain, and he was telling me, I make a survey, a bit like Barker. I make a survey and I look at what the foals eat and all their conditions in my area in Belgium, and I want to see how it influences osteochondrosis. And I told him, no, You have not started? And he said, no. So can you actually put into your model if the mares were fed concentrates or not? And that's what happened. In his study, epidemiological studies, he had an incidence of 33 to 38% of OCD when the mares were fed concentrates during gestation, compared to only 4% where the mares were fed only forage during gestation. And that started a long time collaboration with them because it was really striking. So we actually did it. We did it again in horses. I'm just wondering. So we thought. So I will have to summarize here. You won't have always the real results because it's really a lot of work. So we started to work on the use and the effect of starch, which is really changing the glucose homeostasis in pregnant mares, and see whether it was increasing the risk of developing osteochondrosis. And in experimental setup, we actually showed, it was the end of the thesis of Pauline, that we had an increased risk of developing osteochondrosis when we fed our mares with concentrates during gestation. But the problem is, when we see that what Didier Certain in Belgium did, he asked the owners, did you or not use concentrates? We had no quantity and no quality. Here we used barley and crushed barley, but we couldn't say this is more, this is the limit that you need to go through. And also, a lot of people, when we started to show this type of work, they said, well, I don't have enough hay. I don't have enough access to hay. There's a lot of people who have 1 or 2 horses. They don't have access to hay. They cannot buy enough hay, so they have to use concentrates. So what if I cannot meet the requirements of my horse only with forage? I need to use concentrates, and what should I use? So we have to work with that and try to actually help breeders to actually use the right amount of concentrate. So that was the subject of the second PhD student, who was Morgan Robles. And so, again, this is the summary of a lot of years of work, because we make 1 experiment a year. We breed mares and we decide on the experiment. And so here we had a flattened barley, we started the flattened barley we used it for the last 4.5 months of gestation, and we usually give 1 kilo, so it's not very much to big mares, 1.5 kilo a mare morning and evening. So here we have the group that does not have concentrates, the group that did get concentrates, which we could relate to the starch in the meal. And here's the osteochondrosis in the foals. We have 17% in the controls with just forage, 45% in the foals that got osteochondrosis. So that was confirmed. And I talked to people also in Brazil. Last time I was in a horse meeting, and and I think it might be published, they also have, people show, and you've been showing also an increase in osteochondrosis. So it is solid that this will increase the osteochondrosis. So when we used the flattened barley, and this one also, those are very small numbers, so they need to be verified. But from that, we actually try to think maybe the limit might be around 100 grams starch, based also on work by George and on discussing also with nutritionists from the United States, in particularly Bart Stanyard. And here we have the comparison with maximum 75 grams of starch, so some concentrates but not too much, and minimum, so it might be higher, 110 grams, and you see that in the minimum starch, where we have a lot of starch, we have 80% lesions compared to 20% in the other group. So definitely overnutrition is a problem. And excess starch is a problem, so we need to control the amount of starch. But then we were wondering, what about undernutrition? It could be also a problem. You know, I discussed with you yesterday, and you say, well, you know, once the mare is pregnant, we put her in the field and we don't look at her until term. So they can be undernourished. We just thought that, at least in Europe, we never put them under very strong undernutrition. So we started on 80% of the needs. So Morgan looked at placental vascular adaptation. So we had an increase in vascularization of the placenta, increase in amino acid and vitamin transport. So this placenta was compensating for this undernutrition, which means that we found absolutely no effect of undernutrition on the foal. So if I am a breeder and I see my mare is slightly undernourished, but not too much, and I look at my foal, it looks perfectly healthy, its growth is normal, so is there, however, an effect? And we're not the only ones who showed that. We followed our horses, and in the experimental farm, they were gelding the males at 1 year of age. So I said, yes, I want the testicles, of course. So we looked at the testicles, and they had delayed testicular maturation in the ones that were undernourished. They had reduced insulin sensitivity, that means increased insulin resistance, at 19 months of age. And again, they had reduced cannon width from 19 to 24 months of age when we ended the study. Again, if you're breeding show jumpers, you want their muscle to be working well, you want them to be not resistant to insulin, and you want to have big bones. So again, you know, it's like undernutrition. They may look normal here, but they may pay the price later on. Twink did the same, except when he did his experiment, then he had a severe, he had Streptococcus equi in his herd, so all the mares stopped eating and lost a lot of weight. So he had a severe undernutrition, and yes, with a severe undernutrition, he observed a difference in birth weight. And he observed an increase in the size of the placenta, but the volume of the placenta was decreased, the decreased surface of the microcotyledons. Basically, this placenta was not able to compensate for a very severe undernutrition. It could for a small undernutrition. The owner doesn't see anything, but if it's severe, you will see something. But he didn't look at the foals afterwards, so we don't know what happened. So the other problem that we have, and that's why it's very interesting to talk to you, and because you have the same problem with the brioches, which have a tendency to be a bit obese or very obese. So obesity, we have been discussing, there's overnutrition during pregnancy, where they might have a high body mass index at the end of gestation, but they're normal at the beginning, and there's obesity that is already present by the time you breed the mares. So that was a question that we asked ourselves, and we didn't want to go into the pathology. We didn't want to have animals that were pathologic. We just wanted to have a normal endocrine obesity. So basically, I didn't show it here, but for us, the definition of an obese mare is a mare that has a high BMI. So in France, we use 1 to 5, so above 4. So that would be 8, I guess, for you, above 8, throughout the year. Because usually in winter they lose weight, and then when they go on pasture, then they put on weight, and that's normal. And so a normal mare will go tack, 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 like that. When we define obesity, we see that we don't see the loss. They stay fat. So that's what we did. And so what we found is obesity from early gestation is associated in general with insulin resistance and low-grade inflammation. So we didn't do that. That's other groups. In early gestation, they showed that in the endometrium you have inflammation and expression of inflammatory cytokines, altered lipid regulation, and mitochondrial stress. And in the embryos at 8 and 16 days, they have inflammation, lipid regulation is affected, ER stress, oxidative and mitochondrial stress. Of course, you know, what we say is obesity doesn't really affect fertility, but these embryos are affected. So they will go to term, but they are affected. So then we looked at the whole gestation. So we found placental adaptation in response to obesity. And we looked, and as I told you, and it's back to the early graphs that I showed you, there's also this neonatal period that is really important, and the milk, and you can change things. There's still epigenetically plastic. So we looked at the milk quality in the mares. So everybody was in the same pasture, and even though they were in the same pasture, the mares that were obese had more pro-inflammatory profiles. We didn't see anything on the proteins or lactose, or it was just on the fatty acids, and they have more pro-inflammatory profiles in their milk. When the foals are born, again, I am a breeder, my foal looks absolutely normal, so I don't worry. However, we found an increase in low-grade inflammation until 6 months of age. We used the SAA to measure that. We found an increase in insulin resistance both at 6 and 18 months of age, and an increase in osteochondrosis at 12 months. So obesity will have effects, but more in the long run. So basically, both under and overnutrition alter the long-term offspring health, at least until 2 years of age. And what's really interesting is there's a number of papers now after all this work, because when we did the work, we were the only ones, and it was on our saddlebred herd, and you're always a bit frightened that people will not see the same thing as you, but now, you know, there's quite a lot of work where people find the same type of things. And it really changes how we teach in France now, how we teach breeders. I work, all these grants are for the Institute of the Horse, and the Institute of the Horse is in charge of giving instructions to breeders on how they take care of their horses. And so we have totally changed the way we actually teach nutrition of mares in horses. And we really try to reduce concentrates, increase the really high quality forage and things like that. Anyway, during all these years, we had, so I told you, an experiment a year, and sometimes we had to have both primiparous and multiparous mares. And we had old and young mares. And so, because it's an experimental farm, we didn't have very old mares, but then we started to get bizarre observation. We found that in the mares, the ones that were primiparous, you know, the mares, like any mammal, they become insulin resistant in late gestation. This is normal because to face the very fast growth of the fetus in the last trimester, you need to actually be insulin resistant. Women do, everybody does. Except that so it could be excessive, but here in the primiparous mares, they did not get so much insulin resistant, as if they were not prepared. That was very surprising. That's when we did another experiment where we gave arginine to the mares, and we separated the two groups. And also, they have a reduced placental exchange surface that was also shown by Wilshire and Allen, that was showed at University of Sao Paulo, and we showed it. So we thought, well, these primiparous mares, we might want to have a look at them. What do we know? The literature is plentiful. Primiparous mares produce smaller and lighter foals than the following foals. Lactation, there was very little, and the data is pretty inconsistent. In terms of puberty, still a lot of work saying that still at puberty, they have a reduced size and weight if they are born to primiparous mares. They have a delayed maturation of the glucose metabolism because glucose metabolism will evolve with time in the foals between 6 months and 12 and 18 months of age. And they have delayed testicular maturation. So we started to think we may want to concentrate, and it could be a physiological fetal growth model, growth retardation model. And then we looked at the different types of horses we have in France. So we have the racing mares. They are born, they have their puberty, and they start racing at 3 years of age. They finish racing at 5 years of age, and then they are brood mares. Okay, so they start their first foaling at 5 or 6 years of age. Trotters, their career starts early, but they last longer. So they have their first foals, let's say, before 10. Sports mare, they start later. We start putting them in competition at 3 years of age. They really are considered in the last of the young is 5 years, and then they have a long career. And then only at the end of the career do they become brood mares. So you can have young primiparous mares or old primiparous mares. And so does it make a difference? And then the draft mares, well, they are used for reproduction, so they are, they are used, they wait longer than in the racing, but they start to be bred around here. So we looked at what the literature was telling us. Depending on the breed, about 37 to 63% of the brood mares are more than 10, and 4% are more than 18. In terms of parity, so the primiparous are the ones that fold for the first time. Multiparous fold more than once. And what we found is about 15% of the sport brood mares are bred for the first time at more than 10 years of age. And the average foal production for one mare is 5.5 foals. That means if they start at 11 or 12, they will actually produce foals until quite old. And another question is, if you want to have one foal a year, you're breeding while the mares are nursing. So they don't have the same metabolism as when they're not nursing. And I had a big fight with my manager of the experimental farm because she didn't want to breed the mares every year. And I said, but that's what the industry does. So anyway, so we started to say, well, maybe does lactation at the time of breeding play a role in programming? So the breeders usually aim at one foal per mare per year. And in the literature, between 37 and 65% of the mares, so a wide variation, are in lactation at breeding. So it really varies. So there's in the use of horses, and I don't have the data for career shows, but there's a large variability in the age, the parity, and the metabolic status in normal brood mares. So here is the third PhD student on this series, Emily. So yes, and so we looked at data. So the performance and winning prices in racing are positively correlated to size at 1 year of age, but we showed that the primiparous mares have smaller foals, and the performance is slightly reduced when born to primiparous mares. So I'll show you more of that data later on, but so we know that there is an effect. So Emily, we knew that age, parity, and lactation could play a role. It could play a role on the embryo, on the placenta, on the lactation, or on the foal. And the objective of Emily's thesis was to distinguish what was the effect of age, what was the effect of parity, and what was the effect of lactation. It was a bit too much. She's finished her PhD without finishing, and was still trying to finish the work. We have all the data and all the samples, but it's now the PhD student is gone, so it's difficult. So one take-home message: never give a too big a PhD study to your students, otherwise after it's hard for you. Anyway, so the first thing is we had to decide what old is old. Is old over 15, which is the thing that are said, especially for when there's really a big drop in fertility, or is old not so old? And I work also with humans. And old in humans starts to be, for fertility, starts at 35. The fertility starts to really drop at 35 years of age, and at 40, you have a really low fertility. So we thought, well, 40 is 15 years of age for a mare, let's go a bit earlier. So we looked at the literature, and there are only 2 large studies showing the fertility in mares. And there's the Jeffcott et al. 1982 and Langlois et al. 2004. And you see that there's a slow decrease. It's not like in women. There's a slow decrease in fertility in mares. So there's no hard and fast rule for the age at which mares will experience a decline in fertility. It varies amongst individuals, farms, and locales. But age is still one of the most consistent factors dictating a mare's reproductive capacity. And basically, we kind of felt like, in the papers, they were mentioning 8 or 10 to be really the time when it was decreasing. So that's what we try to look, it really decreases significantly between, So here we start. We have Langlois and Boin in 2004. They used more than 500,000 breedings on several breeds, and they said that mares older than 8 should not be used. These are geneticists. And so because there's the decrease of fertility is more than 10%. And we know there's a significant decrease in pregnancy rate at 42 days by 8 years of age. Between 9 and 14, there's an increase in early embryonic death, a decrease in live-full rates, and then afterwards we also have a decrease in pregnancy rate at 15 to 21 days. So we thought our time for age was around here, and so we used the 10-year threshold. We said mares here are old and mares here are young. So that's why you have to be careful, because my definition of old is not what is used by everybody. We also looked at the literature about parity and nursing. We actually did an epidemiological study that we still have to publish. We compared nursing mares and nulliparous mares, and the nulliparous were very fertile, and the nursing mares didn't seem to have too much of an effect on this fertility. But in lots of the papers, and you see there's a lot of papers, people they compare things that should not be compared. They compared full heat and not full heat, different stages of lactation. So how can you say? So, again, you have the probability for embryo mortality with age of mares. So it was not so clear. And what does it do if we have mares that are older than 10 years? Is it good or not to be an old mare that is nulliparous, or is it better to be a nursing mare and having had a foal already? So we decided to not use full heat. We had data saying that all nulliparous mares seem to be less fertile, but there was no study on the interaction between age and fertility and parity. And there are studies on stud farms in thoroughbreds, but they are different. They don't do the same things. And then in sport mares, we're using AI, which they don't in thoroughbreds, so it's a bit complicated. So, because in France we use artificial insemination. So we did our study in an insemination center in France. So we had 27 of the mares that were nursing. 20% of them were old. We had 65% that were more than 10 years old. And out of those, we had 67% that were both old and nulliparous. And we had 34% that were nulliparous. And we looked at the effects on persisting breeding accumulation and pregnancy rate. So in the old mares, we have an increase in, and you all know, of persisting post-breeding fluid accumulation. You all know, it's not published because one of the reviewers said to us, we all know, but there's no paper actually saying it, showing it with, we all know clinically, but it's not really being shown. And so we have an odd ratio that is 0.4, minus 0.4, to have a pregnancy in old mares, which is okay with new. When they are nulliparous, it's minus 0.74, so it seems that it's a tiny bit worse. When the nulliparous mares are old, then we also have a detrimental effect, 0.57, more or less the same. And then if they are nursing, we have a decrease. Here it's a decrease in the incidence of persisting post-breeding fluid accumulation, which seems logical if they have oxytocin due to the nursing. So we wanted to look at these mechanisms. So anyway, so this is what the background with Emily when she started her PhD. So first we had an experiment between primiparous and multiparous, and we knew that there was a decrease in burst size. Sorry, I'm just wondering whether I put the... Okay, so I didn't put the experimental protocol. It must be later on in the presentation. So we confirmed what we knew, and we confirmed in the placenta we had, when the animals were primiparous, we had a decreased gestation length. Foals were, the placentas were smaller in size, like the foals, less efficient, but otherwise the structure was pretty similar, and very few genes were altered, and they were all on feto-maternal communication. Then we looked at embryos. We collected embryos from primiparous mares and multiparous mares of the same age group, young mares. And we saw that the embryos from primiparous mares had a slight alteration in gene expression. And again, it was in feto-maternal communication. So as if, again, like what we saw in the primiparous mare when they're pregnant, it's the embryo-maternal communication that is not totally adapted, and that's why it's different. Then we looked at the foals. Everybody caught up. And we looked at the milk, and the milk production was decreased in the primiparous mares, but the quality of the milk and the energy brought by the milk was increased, which actually really compensated. So those foals grew absolutely normally and had no difference in osteochondrosis, nor glucose metabolism. So then we looked at the effect of age. So again, we know fertility lowers with age and embryo loss increases with age. What did we have in the literature? There was papers saying that the mitochondrial quality of the oocyte is decreased. It's mainly the group of Colorado that works on that. And they are susceptible to poor chromosome alignment due to instability of the achromatic spindle. So what happens if the embryo produces a foal? So we had... Again, we have in the literature that the mare age will affect the size of the foals. The foals are smaller at birth in very young mares and very old mares, and they are smaller and lighter also after 510 days in very old mares. That's only in one paper. It's a very old paper of 79. And what we had seen in the literature also is that in young mares, the maternal metabolism was altered with plasma osteocalcin and parathyroid hormones that were altered, but we didn't know exactly what it meant. What we had also with age, that the group of twink, but others, showed an altered vascularization of the placenta during gestation in older mares as mare aged, and same placental weight but structural alteration. That was also the group of Allen and also the group of Mereyes in Brazil. And in terms of milk, there was very few papers, and they said that they had similar quantity and quality. But nursing, yes, insemination could be a confounding factor. So here's our experimental plan. Here it is. So we have 2 farms. Chambray. We compared young nulliparous to young multiparous, and we compared the effect of parity, which I showed you. Then we had, in the same herd, we had young multiparous that were between 4, 5, and 6 mostly, and old multiparous. And we could compare effect of age. I have to tell you that we always use 1 stallion per year. So when I compare groups, they have been bred by the same stallion. And then we have nursing old multiparous and old multiparous that are not nursing, so we could compare the effect of nursing. And then in the Haras du Pin farm, we had old nulliparous and old multiparous. We didn't have any old nulliparous in this one because this one is really for reproduction. So we looked at the effect of parity in old mares. And so we collected uterine fluids, but I won't tell you about the uterine fluids. They've all been analyzed, but I don't know what the results. I need to find someone to help me to analyze. So we did proteomics, we did lipidomics, etc., but I don't have the results. And we collected the embryos. In most of the mares, we couldn't do that in this group for practical reasons. Then we collected the placenta, we monitored lactation until weaning, we followed up the foals, same thing, osteochondrosis and glucose metabolism until 18 months of age. And also we made sure that foals were born as much as possible in April, so that they were from March to May, but the herd was mostly in April, so that we don't have an effect of season. So the effect of parity. So here we have the young multiparous and the young nulliparous. We collect, so we do a usual protocol. We induce ovulation with HCG, we do AI, then we check for ovulation, and we collect the embryos at 8 days of age. So that's just the growth, and so we collect the blastocyst. So, one of the things is, as I told you, the blastocyst is made of the ICM, which will make the foal, and trophoblast, that will make the placenta. But if you just analyze a blastocyst, you have both mixed. We are lucky in horses because we have large embryos. So we actually cut the embryos in 2. So we had one part with ICM and trophoblast and one part only with trophoblast. And in cancer research, in order to look at gene expression in the cancer cells and the non-cancer cells, they usually have mixed cancer cells. And they do exactly the same thing. They actually use a technique called deconvolution, which allows you to actually conclude what is the expression in the cells of interest compared to the ones that are pure here, which is what we did. So we could actually have the ICM and the trophoblast. So in the ICM, these are the young, so we are comparing the young nulliparous to the multiparous, and there is not 13,000 genes are expressed, but not so many differences in gene expression, either up or down regulated, but not very much difference between the nulliparous and the multiparous. However, when we see here in the upregulation, it's genes that are related to development and morphogenesis of the embryo. And in the downregulated, in the nulliparous, you have regulation of transcription, cell differentiation, and metabolism of hydrogen peroxide, so stress. So that might be still playing a role. And then we found also in the trophoblast, we found different things, upregulation of iron-binding proteins, response to hypoxia, and prostaglandin F2 alpha, which is and prostamide, and amino acid transport. So that's really related to placental function. So there are differences already at that age, not very much, but there are differences. So we can conclude that between nulliparous young males and multiparous, you have slight alteration of gene expression in cell metabolism, embryo development, and signaling and transport. Then, when you do these type of experiments, sorry, this is a very heavy graph, you actually first look at the genes that are differentially regulated, but it can be one gene because the tests are very stringent. So then afterwards, you use another bioinformatic thing to look at what genes, where these genes belong to. Do they belong to two groups of genes that have a significance in terms of biology? So this is what we did. This is called GSEA, gene set evaluation analysis. And so in a cell mass, a lot of these genes were also related to RNA production and maturation for the young multiparous, where in the trophoblast, we had fatty acid transport and oxidation, interleukin signaling, and energy metabolism and regulation in the trophoblast that were affected. These pathways were affected. So we have, again, what we saw before that I summarized, differences in embryo-maternal communication long before implantation, especially with these, in those embryos. So then we looked at the placenta. So the placentas are collected. We actually look at the growth surface, and then we do analysis in terms of RNA-seq and histology. So as I told you, it's an epithelial placenta, so we get these, and we actually measure the different types of cells, of trophoblastic cells, fetal vessels, and the uterine cells, and we look at the interface here. So that's what Elisa has been doing quite a lot. So anyway, when you have young primiparous and young multiparous, well, the young primiparous have longer gestation lengths compared to multiparous. However, the placental weight is smaller and the fall weight is smaller. And the placental efficiency is reduced. We didn't find any difference in the structure, and we have a reduced placental size without changes. In terms of gene expression, only 2 genes were differentially expressed. The free fatty acid receptor, it's involved in the regulation of inflammation, and the interleukin-4 receptor involved also in inflammation. And it was increased in the primiparous males. In terms of pathways in those placentas, we have gene sets related to inflammation and immune response. So we don't understand really why in primiparous males you have more immune response, maybe because they are not yet ready, and compared to multiparous, and gene sets related to cyclic nucleotides. So a few changes in gene expression that could indicate alteration in fetal-maternal communication in primiparous compared to multiparous. Then we looked at the falls, and you see we don't see really a difference between the 2 groups. They start to be smaller at birth, but they catch up completely afterwards. You see here it's significant, and then afterwards, no difference. I don't even mention the osteochondrosis because our friends from Liège, they went here for 2 years. They came for nothing. They found no difference between any of the groups. And the milk, it's just the energy concentration in the multiparous is smaller than the primiparous. So the primiparous have a richer milk, and the yield, however, is smaller. So in conclusion, for the effect of primiparity, that's what you saw. We have a reduce in gestation, an increase in gestation length, efficiency that is reduced. We have some intrauterine growth retardation. The embryo are slightly altered, but afterwards they catch up, and you don't really have to worry. The milk production is enough to make them grow. So aging, we did the same. So in the embryos, you see that we have much more genes that are differentially expressed compared to parity, especially downregulated in the inner cell mass. And what was really interesting in the old mares, I mean, the cell cycle, mitotic division, cell signaling, transcription, embryo development, and mitochondrial function is altered in the old mares. And in trophoblast, we find, again, mitochondrial function, protein catabolism, translation, embryo development. So a lot of genes are altered in the old mares compared to the young mares. And that actually fits with what we know is fertility is decreasing, and probably these embryos are disturbed, and that's why they don't survive. In terms of gestation, the old mares and the young mares, so the old mares had longer gestation lengths. There was no difference in the morphology of placenta. However, when we looked at the structure of the placenta, the young mares, sorry for changing here is green, it's the young, and yellow is the old. So the surface of the allantoic vessels is reduced, and the allantoic vessels are the big vessels that go to the placenta and bring the nutriment to the placenta. In compensation, there's an increase in connective tissue in the microcotyledons, the exchange area, and in the total volume, but we're not sure this is totally compensatory. So the main point was the surface of the allantoic vessels was reduced in the old ones. And in terms of genes, we had several genes that were disturbed. And basically, in the old mares, we had regulation of inflammation and immunity, transcription, cell adhesion, and ionic homeostasis. And we're really wondering, this regulation of inflammation and immunity in old mares may be due to the fact that the uterus is not as good. And that's why the uterus is not, so that's in the placenta. So in terms of foal lactation, we didn't find any difference in the foals, whether they were born from old or young. No difference in milk production, and except for the protein concentration that was slightly higher, that was higher at birth, but it's 3 days after birth. And so for the effects of aging, we know that the embryos are altered, which may be at the origin of infertility. The gestation length is increased, but they are altered gene expression, which may be due to uterus degenerative changes. But this placenta adapts, and there's no difference at birth. There's no difference in milk, and these animals just do well. So basically, if you're born from an old mare and you pass this embryo time, that is really the tough time, you're likely to be totally okay, which was an interesting one. And so this plays for maybe using antioxidant or mitochondrial stimulants as supplementary things for the mares. That's what the people in Colorado do for oocytes. And before I started to recommend that, we did another experiment that is not published yet, but should be soon, where we did another experiment where we again collected embryos from old mares and young mares and then really focused on mitochondrial function. And yes, there is effect on mitochondrial function. Oh yes, and I didn't show here, but in late, so the question was that enough for foal growth? But we didn't see any difference. So the main point afterwards was, what if you're old and nulliparous, or an old and multiparous, and is it bad? So we had only the embryos because we couldn't foal those mares in the experimental farm. We found differentially expressed genes, and they were affecting very few gene sets affected, and but there was more effects on the old nulliparous compared to the old multiparous. So we feel that it's even worse to be an old nulliparous because you have the effect of being nulliparous and being old. especially the genes here that ApoB is really important for lipid metabolism, and it's one of the key genes, and AOMS is a really very important gene also for development. So, I mean, those genes are really very key genes, so we are really wondering whether it's, it's really important in old nulliparous mares. So the conclusion for the cumulative effects is that there is an alteration of gene expression in the embryos from old nulliparous with effects on embryo development and transport of molecules, maybe due to uterine degenerative changes and priming of the uterus, but we don't know. And that may be at the origin of the decrease in fertility, and we have no idea of long-term effects. So the last group, I'm sorry, it's long, but I hope you can follow, is the lactation. So we couldn't get the embryos for lactation, but we did the monitoring, the placenta collection, and we followed up everything. So the nursing ones have a smaller gestation length compared to the they are nursing old multiparous compared to old multiparous. So the nursing have a shorter gestation length. We didn't see any difference in the placenta in terms of structure. In terms of gene expression, we found quite a lot of genes differentially expressed, and they were really related to inflammation and immunity and cell division, but only a few here. So there is an effect on inflammation and immunity for being non-nursing compared to nursing, and protein synthesis. When we looked at the foals, you see the foals grew well, but the foals were smaller at 480 days. And when we looked at the milk yield of these nursing mares, the milk yield was not different at the beginning of lactation, but was really smaller at the end of lactation. So this difference might be due to a late difference here that we are not yet significant, due to the poor lactation at the end of lactation of mares that were bred when they were nursing. So the idea here is that probably when we are breeding mares every year, we really have to be careful at the end of lactation to supplement foals in time and follow the growth and supplement them before they are weaned, because the mares may not be able to produce enough milk to go up to weaning. So basically, very effects on the placental development and immunity. That might be due to changes in maternal nutriment availability in the nursing mares. No difference in birth morphology, then no difference in growth, but a reduced postnatal growth at the end of weaning, and reduced production of milk. But the quality was the same. So, last one. So my late husband was always working with me, and he was like, it's interesting what you do, but what is important is performance. If you don't have performance, what you do is absolutely useless. So we finally did it. In France, we're very lucky. We have the Institute of the Horse, and every performance of every show jumping in France is registered, not only in terms of size of the hurdles, but also in terms of position in the competition. And so we have this data for 25 years. And at the same time, we have the genetics of everybody. We don't have the full genomic data, but we have the genetics. We know if it was the first foal, the second foal, So here we are. Morgan is still here, and we say, you're going to do a postdoc with us, and you're going to do data analysis. So we use the database with a geneticist called Anne Ricard. And here is the data. So we have the genomic level, the genetic level of each horse. A genetic level is reassessed every year. So 100 is the mean of the population. If you're more than 100, you are good for breeding. You are going to bring genetic progress. The best horses are 140, 150, 160. And if you're less than 100, you should not breed for them if you want to have genetic progress. So we monitor that, but here I had the whole population. So we had to adjust all our data to the genetic value of the horses adjusted every year. So it was quite a long work. And with a geneticist who said, epigenetics is not strong enough, I don't care. So, but we went on. Here we have adjusted for genetics. So there's no genetic effects. And we are looking at performance and in the different animals. So those that were born to 3 to 4 years old mares, of course they were primiparous, they have a slight increase, but really not very much. And you see the bar is going over the 0, so it's not statistically significant, of their performance compared to their genetics. When the mares are 5 to 10 years old and they have their first foal, and so they are not lactating, the fact to be born from a 5 to 10 years old primiparous mares not lactating will reduce your performance as a foal and as an adult, as an offspring. If the mares is at her 2nd to 5th pregnancy, if she's not lactating, again, the performance is reduced regarding to the genetics, to the expected genetics. But if she's lactating, you are exactly at the right level according to your genetics. Then if we looked at older mares, again, you have the ones that were first parity, not lactating, buff. Again, the performance is lower than expected due to the genetics. If these horses are born from the 2nd to 5th lactation of non-lactating mares, it's slightly increased, but again, not significantly. But if the mares are lactating, the performance is increased. If they are born to 6 years old or plus mares, there's not very much difference. It seems that born to be 6 years old or more is good, regardless of genetic quality. And you see that there's not very much difference whether the mares were lactating or not at the time of breeding. So this was actually confirming that primiparity is not good for the performance in show jumping. What was interesting is that age mares, they can actually produce animals that will go over their genetic potential. And the question was here, lactation seems to be a positive thing for performance, that the fact that the mares fall every year is definitely not a problem, and they actually do better. And here, our interpretation was that regardless of genetics, if breeders use mares to breed at 3 years old, it's because they have a fantastic genetic. So they will really invest in all the other environmental factors. And that's why we have a slight increase. That was our interpretation. So we don't see the effect of primiparity because they really try to improve the environment. So in conclusion, in the effect for embryos, effect of parity, we have an alteration of the inner cell mass growth, alteration of the trophoblast function. It might be that the embryo takes time to adapt to a different environment, or it may play a role in maternal recognition of pregnancy. In terms of maternal age, we have delayed and poor embryo development in the old multiparous, coherent with other studies on oocytes and embryos, which may explain the embryo loss. In terms of parity in old mares, we have an alteration in embryo development of trophoblast function, lipid metabolism. So it might be that to be an old nulliparous is worse than being an old multiparous. In terms of placenta, in terms of parity, the placentas, they are smaller, but they are really similar, and the foals catch up, and maybe due to better quality of the milk. In embryos, we had a disturbed trophoblast that could alter placenta development, but then afterwards it went well. In terms of multiparity between the old and the young, there are different placentas, but no alteration of foal development. So maybe these placentas are totally able to compensate. And then in terms of suckling, we have an alteration of placental function. These placenta seems to adapt to a different metabolism. And however, we didn't see anything really on the foals, but we stopped at 1.5 years of age. And however, before weaning, we had a problem of lactation. So I think I will go to more the summary of. So we know that. Do I have a better? Sorry, I'm just looking. I don't remember the end of. Yeah, I have the take-home message afterwards, which is going to be the most important. So in summary of all I told you, maternal quality feeding throughout pregnancy is important. You should reduce concentrates in mare's feed, but keep body condition score at around 5 in English system, 3 to 3.5 in our French system. Definitely avoid obesity. Primiparous mares are smaller foals, so it's an important thing. One of the things we are thinking with jumping mares is that if they really have very good genetics, it might be good to actually make them have a foal before their career and then have them go on with their career. And the next experiment is going to be, we're going to exercise the mares during pregnancy, so we'll see, to see what it does. We didn't see any obvious deleterious effects of reasonable mare aging on the quality of the foals, so we can go on, except that we made also another paper with Eric showing that if you breed old mares, you're losing the genetic progress. So he thought it was completely stupid to breed old mares. You can breed yearly, but be prepared to support the full growth before weaning. And embryo transfer, the choice of the recipient is really important. And we've put that in vet clinics of North America if you are interested, with more details also on the food. So the take-home message, so when we see the breeders, we say avoid breeding old mares, but more for loss of fertility and loss of genetics, but otherwise the old mares will produce good animals. Adapt the reproductive strategy. They all want to use frozen semen. Well, it might be better because they're less fertile to use fresh or refrigerated semen, or use embryo transfer, or or in vitro production, because the oocytes can be damaged. In terms of parity, it might be important to breed them as early as possible to avoid the cumulative effect of nulliparity and old mares, so especially in show jumping. In terms of nursing, I think I showed you for fertility, it's better to breed the nursing mares, probably due to oxytocin. The fluid accumulation is less in nursing mares. For postnatal development, you have to be careful about the lactation. And you have to provide a nutrition that is optimal to reduce the cumulative effects of lactation and gestation, and we need more work. And that's it. And so what we need now, we are totally underestimated the effects of lots of things. So, we need to be a big community to work on this, to understand and to help breeders, because, you still have the genetics that are doing a lot of the effects, but how we treat the animals are important. We are starting with University of Dublin studies on stallions, because we know that in males, in other species, they do program. So now we are starting the male way. And a lot of stallions are obese. We might have problems. And we don't have any markers. I had a hard time to get money to do epigenetics, because my institute says, I want you to give things for the breeders. I don't care about the mechanisms. Finally, they funded it this last year, so we will start having mechanisms. But It's difficult. We don't have a marker to say, oh, I do a blood sample to your mare. Your mare is at risk or not. At the moment, we have nothing. We tried a lot of things and nothing works. So that's a problem, and it's expensive to develop. And why is it important? Because when I was at school, I learned that phenotype is the effect of genotype plus genetics plus the environment, effect of the environment. However, with Eric, we showed that the genetic value for performance in show jumping explains 24% of the performance. The genetic maternal value, which is the uterus of the mare, really, 1.4%. The permanent environment of the horse explains 24%. The common environment of the offspring and the same mare, 1.6%, that's for embryo transfer. The residual for performance is almost 50%. So that means when you select genetically, you only select for 24% of the performance, at least in show jumping. With your environment, you can improve the performance by about 25%, but you still have this unknown. That's where epigenetics come in. And they're really difficult to study. You saw all the mechanisms. In most cases, people do methylation. They don't look at histone acetylation and mRNA, microRNA. So it's really a problem because I've never, in any species, I've never seen a study where they study the 3 epigenetic mechanisms at the same time. So when I talk to epigeneticists about that, they say, oh yes, but it goes the same way. And I say, are you sure? Okay, so we have the interactions. The genetics will drive the microRNAs. The epigenetics, the environment will drive the epigenetics, and the epigenetics will drive some of those. So we are in trouble. And now we know that the metagenomics are playing a role. And it's also one line that would be important to do. We know in rodents that microbiota are different between obese and not obese mares, not obese females, and that if you replace the microbiota, you can actually program the newborns, and it's really a way we could act on the newborn. But it's a lot of work, again, and it's a totally unknown field. So voilà, and this took 15 years, and we could collaborate with a lot of people, and I hope we can add your University of Montevideo to this list. Thank you, and I'm ready to answer any questions.